I'll make all those cancer treatment top hat things as homework. We're going to start the next bit about meiosis and genetics, but we're going to first go over some of the stuff we already learned. So, the cells in your body that do give rise to sperm and eggs are called what? Basically happen. Okay, so if we look on the bottom here, there's three answers for three of these different questions: enzymal, anchorage dependence, cell cycle control, contact inhibition. Cancer cells differ from normal cells because they divide when they shouldn't, and that's loss of what? Invading, loss of what allows invading surrounding tissue, contact inhibition, or anchorage dependence? What do you think? Contact inhibition or anchorage dependence? Huh? Yeah. And then obviously, metastatic cancer, loss of anchorage dependence. Remember I talked about radiation therapy, it damages the cells so that the tumor suppressor system actually recognizes damaged cells and causes those cells to undergo apoptosis. So which of these? Radiation therapy is least effective against contact inhibition, anchorage dependence, in other words, invasive cancer, metastatic cancer, or causing cells to kill themselves. Anybody? Yeah. Why is it good to take multiple biopsies from a single tumor? Because unregulated cell division leads to increased genetic diversity. Remember, cells are dividing every once in a while, a new mutation starts. Yeah. Because a given drug may be effective against only one part of a tumor with one kind of mutation. Yeah. Because tumor evolution can occur, obviously it is. Keep dividing, the more likely it is they're going to develop unregulated cell growth because of mutations, or it should be D, all of these are true. That. Okay. Okay, so here's the cell cycle. Remember, G1, most cells are in G1 98% of their lives. Some, all of their lives, like neurons. Once they're there, they don't continually replicate. Whereas other cells, like skin cells, are more likely to go into the rest of interphase. If a cell is going to divide and replicate, it needs a signal first, which it gets in G1. That institutes or causes the synthesis of all the DNA. So there's now two full copies of the genome in this cell. cells need energy to replicate. You're not just replicating the DNA, but all the other things in a cell, including mitochondria, translational machinery, all kinds of different things. So a cell has to grow enough and have enough nutrients so that it can actually undergo replication successfully. Otherwise, it would start to replicate and then it wouldn't make it because it wouldn't have enough resources. And then finally, the actual replication into two different cells starts with mitosis. And there's 4 major phases. Prophase, where the chromosomes or the DNA condenses in the chromosomes. Metaphase, where all these chromosomes that are not duplicated line up along the middle of the cell. There are these microtubules that come down and connect to these chromosomes and then start pulling apart so that each cell has a full complement of all the chromosomes. And the pulling apart phase is called anaphase. And then nuclear envelope starts to reform. So in prophase, the nuclear envelope starts to break down, which is where all the DNA is, and that condenses. And then telophase is where it reforms, and then cytokinesis is finally where you have 2 daughter cells that are identical to each other unless there's any sort of mutations. CP. Checkpoint, and then MCP. Okay, so there's three checkpoints. One in G1, after a cell has to have the right signal to replicate. If it starts replicating without that signal, the tumor suppressor system kicks in. G2 the cell has to be large enough. All the DNA has to be double the amount. And then in mitosis, the chromosomes have to separate equally. So each side gets homologous chromosomes instead of one getting 1 chromosome and the other getting 3. Okay, we'll stop there. And